Okay, so I'm going to start with the presentation. So thank you everybody to be there today. So it's a monthly webinar from EUROTOX, as some of you are aficionados of this webinar, so I can see the names. Today I'm very pleased to have Jan Willem van der Laan, because Jan Willem van der Laan is a very well-recognized regulatory toxicologist. So since May 2024, Dr. Jan Willem van der Laan is fully retired as a senior assessor in pharmacology and toxicology of the Medicine Evaluation Board, located in Utrecht in the Netherlands. On behalf of this board, he was for a long period a member of the Safety Working Party for EMA, and he contributed a lot on the Committee on Human Medicinal Products, and he was the chair of the Safety Working Party at EMA from 2012 to 2021. So he's also highly involved with the International Conference on Harmonization, the ICH process, started in early 1992, when he became a member of the expert working group on carcinogenicity testing. Later, in 2005, he was a member of regulatory rapporteur on immunotoxicity testing with the ICH S8 guideline. In 2011, he finished as a final rapporteur the addendum of the preclinical testing of biotechnology derived protein. And so the S6, he was a member and regulatory chair of the expert working group on carcinogenicity testing, starting in 2011-2012, finishing in the summer of 2022 in Athens, releasing the new version of the S1B SCH guideline on carcinogenicity. So this new version is providing a national approach on carcinogenicity testing, circumventing the 2-year study in rats. And so today, Jan Willem accepted to give us a presentation of this on a reduction of animal use in carcinogenicity testing of human pharmaceuticals, which is a very timely topic in this field. So thank you very much, Jan Willem, and the floor is yours.
Okay, dear audience, dear colleagues, thanks to Mark and the EUROTOX to invite me in this honorable presentation on the reduction of animal use in carcinogenicity testing of human pharmaceuticals. It's important also to be aware that is the restriction, that it is on human pharmaceuticals and not on chemicals in general. Those developments are more generalized later on. So we start just fundamentally with the ideas of cancer. If we have in mind the safety of human pharmaceuticals, we should focus on the safety also with respect to the induction of cancer. There is a DNA damage response. There's a growth advantage in cancer cells, so we have to think about all the genes that are involved in that process. But then if there is a proliferation of cells, then there will be also an invasion of cells into neighboring tissues, and the survival of these disseminated cells, which can be interpreted as metastasis. And all those processes are involved in carcinogenicity testing, although there are some restrictions scientifically. The authors Hannegan and Weinberg have presented already for 3 times now the hallmarks of cancer, the first time in 2000, and later on with an update in 2011 and 2022, indicating that there are a lot of factors involved in the induction of cancer. And if we are testing for cancer, we have to be aware of all these factors, although we cannot all test testing them. So we focus on the sustained proliferative signaling. We think about growth suppressors, avoiding immune destruction, but resisting cell death might be important. But all these factors together are included in the live testing program. But we should be aware of them if we are thinking about reduction of animal use. So the classical approach for carcinogenicity testing is that late in development for human pharmaceuticals, which is parallel to clinical phase 3, we have a lifelong testing in rodents based upon maximum exposure. At least that was the start of the model, in 2 species, rats and mice. And there is a standardized testing with the OECD protocol under good laboratory practice since 1979, and that was the basis for most of the guidelines. So the standard study design consists of 4 groups: a control, low, mid, and high dose, and there are criteria for that. Per group, we have males and females, 50 to 70 per group, with a duration for rats of 104 weeks, 2 years. And for mice, dependent on the strain, it's 78 to 104 weeks. So there is a scheduled early termination if the survival is so that there are not more than 20 to 25 animals. And there are specific statistical types of analysis, including the latency and the number of tumors developed by Dr. Pito. And that is all general knowledge in the literature. If we think about the human pharmaceuticals, we have done some evaluation with my co-worker Justus van Oosterhout and others, and also German colleagues, indicating that from the compounds on the market since 1980 to 1995, the 171, that 50% of them is positive, but still they are on the market as safe drugs. Later on, it's confirmed by Anita Friedrich and Klaus Olejniczak for the period between 1995 to 2009. And they confirmed this statistics as well. And that is important to keep in mind for further discussions. So 50% of all chronically used human pharmaceuticals induce tumors in rodents. However, only 20 human pharmaceutical carcinogens have been identified by epidemiology. And so that's far less than the 170 that are in the previous slide. In the 90s, Ray Tennant from the US developed the concept that the risk of carcinogens would increase if there are multi-species, so positive in rats and mice together, and multi-strain, multi-sex, always in both sexes, and multi-site. However, is that always true for pharmaceuticals? And that was the start of being critical on this type of approaches. So if you think about, for instance, on antipsychotics, they usually induce pancreatic tumors, memory tumors, pituitary gland adenoma and hyperplasia in both rats and mice, but still they are on the market and can be used in the appropriate dose. So these are only a few examples, but there are more. And so is it fully true for human pharmaceuticals that multi is increasing the risk. We have done a lot of work on the guidance of carcinogenicity testing, and we have focused on the mechanism of action as well as an exposure. In my talk, we're focusing on the non-genotoxic compounds, so which are not damaging DNA. That's the genotoxicity area. So the non-genotoxic area is covered in this presentation. Then we have 2-year studies in rodents at high exposure. The classic design indicates the maximum tolerable dose, whatever it may be. In the IGHS1C, we have defined that in general, if a compound is not very potent, that we can have a 25 multiple of the human AUC. In phase 3, the data on human AUC are already present. In some cases, you can rely on the maximum dose to be reached with saturated absorption, and that's also important in this case, a pharmacodynamic endpoint. For instance, with some compounds such as anti-diabetics and so. There are several types of non-genotoxic carcinogenicity, which are important to consider. Also in what is the risk for humans. So there's chronic cell injury, immunosuppression, that's a very relevant one for the human risk. Increased secretion of trophic hormones, I think we can mention luteinizing hormone and prolactin, receptor-mediated mechanisms, and the induction of liver metabolic enzymes. The last one is a high risk factor for rodent carcinogenicity. However, we all agree that the induction of liver metabolic enzymes is hardly relevant for the human situation. So some examples for the increased secretion of trophic hormones, the Leydig cell adenomas by increased luteinizing hormone with finasteride and dopamine agonists. But also the memory tumors, as mentioned earlier, by the increased prolactin. You see positive outcomes for haloperidol, chlorpromazine, olanzapine, the more recent ones, but also the old bromperidol. However, ciprasidone as well as pimoside are negative. So why are not all compounds at risk? I've done at least a study with the GLP-1R agonists, as it was apparent that within the first few, there was so important difference between the studies that I was puzzled. What is the background for that? And we have found out, and we have published that analysis, that the difference between the various GLP-1R agonists are related to receptor potency, but also the duration of the pharmacodynamic stimulation, which is of course also related to the pharmacokinetic half-life. We developed a model based on exenatide and liraglutide, the first ones that were on the market. And we used that to predict the hyperplasia for lixisenatide. So short-acting exenatide with a very low incidence of tumors versus long-acting exenatide, the slow release, with a higher incidence of C-cell hyperplasia. And the same is true for the slow-acting, the short-acting exenatide versus the long-acting liraglutide. So this was the basis for the hypothesis that the duration of the pharmacodynamic activity over the day is important for the risk of hyperplasia and the follow-up of tumors. This is just a summary of the possible consequences of the cell proliferation. So growth factors will increase cell proliferation. Repressors might also lead to a feedback, and that will lead to an increased rate of fixation of spontaneous mutations. That's the general, that's included in this hypothesis. There are focal hyperplasia with altered differentiation, and then this will be related to the establishment of a mutator phenotype, which first will lead to a benign tumor, and then with progressive accumulation of mutations and chromosomal rearrangements, that might lead to a malignant tumor. Cell proliferation is neither necessary nor sufficient for chemical carcinogenicity, but it's still there. There are examples that cell proliferation doesn't contribute, but in most cases it does. So that's the reason that we focus on that proliferation. So in the studies that we have mentioned earlier, van Oosterhout in 1997, and in the same issue, there was a similar paper written by Contreras and Di George to support the writing of the ICH S1B, also to come to more relevant testing. And we concluded there is room for replacing the long-term mouse for other approaches. And in that time, it was in 1995, 1998, we could decide that one long-term rodent carcinogenicity study should be sufficient, plus one other study of the type mentioned in 422, that's listed below. First, a short or medium-term in vivo rodent test system with carcinogenic endpoints. And those are the transgenic mice, well-known, firstly P53, and later on the TGRSH2. Of course, companies are still allowed to do the full mouse study. In fact, the writing of the ICH S6 guideline, which was a parallel process in the ICH, gave us already an indication of new developments in carcinogenicity studies. And then products that have the potential to support or induce proliferation of transformed cells and clonal expansion might be at risk, as in the case for the effects of the compounds in the human body, and this type of effect can be studied in vitro to lead to cause for concern about carcinogenic potential. And then if there is a cause for concern, then further studies in relevant animal models may be needed. And it is important that one of the issues at that time in the S6 was that there was already a recognition that the standard carcinogenicity assays are generally inappropriate. Most of the biopharmaceuticals at that time were species specific and could not be given to rodents and should be tested somewhere else. So a variety of approaches is possible. What are the relevant facts, whether or not it is a growth factor? Is there any immunosuppression? And what is the duration of clinical dosing? So the solution is then that we would rely on the receptor expression on cell lines. and the ability to stimulate growth in normal and tumor cell lines. What we did in later studies, I will come to that later. This strategy could be based on the weight of evidence approach, and this is the first time that the weight of evidence is included. And that weight of evidence approach should include a review of relevant data from a variety of sources. That can include published data. Not all data have to be produced again by companies. So there is information on class effects, detailed information on target biology, in vitro data on the mechanism of action, data from chronic toxicity studies that will be present anyway, and in some cases, clinical data. And that might be sufficient to address carcinogenic potential and inform about the clinical risk without additional non-clinical studies. Be aware that this document for the S6 was present in 2011. The approach indicated that the mechanism of action might raise concern regarding the potential of carcinogenicity. So the pharmacodynamics, so for immunosuppressives, antibodies, and growth factors, the growth factor proteins. And if the weight of evidence supports the concern regarding the carcinogenic potential, then rodent bioassays are not warranted. There's no need to confirm that because there is sufficient evidence present. So in this case, potential hazard can be best addressed by product labeling and risk management practices. When the body of evidence is unclear, the sponsor can propose additional studies, not necessarily the 2-year rat studies or 2-year mouse studies, but it might also be other studies. Just because of this outcome, we did some studies. I have no data, no presentation on that, but you can find it in the literature that the insulin and the insulin glargine has been tested in depth about the role of in carcinogenicity testing, and that's an interesting topic. Please, you can find it in the literature. What we did in 2011 was rethinking the rodent carcinogenicity testing for small molecules. So we have this conclusion on the biopharmaceuticals, and then we go back to the human pharmaceuticals with small molecules, not biotech products. And then we came to the formulation of the S1 regulatory notice document. What is the background of that? In 2011, there were 15 years after writing the S1B, with what I told earlier about the use of the transgenic mice. And there was also the finding from Friedrich and Olejniczak that with regard to the carcinogenicity studies in human pharmaceuticals, nothing has been changed. The pattern was very similar. Most of these studies are not very relevant. The transgenic mice studies are being accepted, but the impact is low. Its use is infrequent, and the answers are doesn't give too much insight. So there's a continuous discussion to reduce the long-term rat study, and then the pharmaceutical industry with Reddy and Sister from the Merck research have put together the results of many compounds, and especially the paper of Sister was leading in this. Chronic tox studies are good predictors of the negative outcome in the 2-year rat carc studies. That was the hypothesis or in the recommendation from the paper of Sister in 2011. I cannot go into all the details, but that's an important. That was the start of a new round for the ICH. So the predictivity of an organ by organ basis is poor. So positive carcinogenicity testing is in fact very difficult to be predicted on the 6 month study, but the negative prediction was much higher. Can we then rely on that analysis on negative tox studies? That was the topic of the ICH in the period afterwards. So the hypothesis is that a rat chronic tox study will identify an effect that would define the need for to complete a 2-year essay. So if there is positive histopathology, then run a 2-year bioassay. But if it is negative, there's no need to perform a 2-year study. Of course, there were some false negatives in this analysis. And what we did, we got all the data from the Sister paper, the background data, and we could identify the compounds because the FDA shared with us the codes, and we could identify the backgrounds that have the pharmaceuticals that have been used. And then if we make that analysis again based on pharmacology, then the false negatives, 24 on that was more than 10% of the whole data set. It could be give some doubt about the conclusions of that analysis. But we concluded that if you go in detail to the compounds, that there's no need to rely on that to indicate that the false negatives will change the outcome of the study. And that data finally will have been published, although again anonymized, in this paper in the Critical Review of Toxicology, together with other European colleagues, as well as Marco Passanen, who joined me in the ICH. And we have in that paper we have included an analysis of 298 compounds from pharma, additional compounds from FDA, and 63 compounds from the Japanese pharmaceutical industry. So, and we came to the conclusion that there are pharmacological classes of compounds, and you see compounds with a number of 13 antipsychotics, for which 9 were positive, but also much smaller classes, vitamin D analogs, with 4 compounds with all 4 positive. Some carcinogenicity was not related to pharmacology, but that was related to the liver effects. But more surprisingly, there was a huge number of classes that were negative. All compounds, for instance, ACE inhibitors, 7 ACE inhibitors, were all negative in carcinogenicity testing. And so there are many more. And also this negative class ID was very important in the analysis. We have repeated that study and that analysis with a data set with a small overlap, but with our old data set published in 1997, including other and more updated compounds as well. We came to the same conclusion. And this led to the evaluation of the possibility to change the need for carcinogenicity studies. So the new hypothesis is that when the outcome is predictable, studies are not needed. We have a positive prediction on the basis of the pharmacology. The positive classes support positive prediction. For instance, the presence of proliferative signals in histopath, hypertrophy and hyperplasia. It's not that you have an organ-to-organ prediction. That was not possible. That was already well known. However, the mechanism of action is still important. And that's why proliferative signals in the pharmacology were important for the prediction. Also, the negative prediction we have tested again, the hypothesis from Sister, and that was all in. And that was also included in the new test, the regulatory notice document. What we did was a prospective study asking companies to submit their CARC studies. So, and then we were asking for the companies to indicate whether to which category they should belong. Category 1 as potentially highly tumorigenic. Category 2, we don't know. Category 3A, tumorigenic in rats but not in humans. And 3B, highly likely not to be tumorigenic in both rats and humans. So in that case, category 3B, a study would not add value. But that's also true for category 1. If there is strong evidence for carcinogenic risk, why confirming that in a rat study if you already know it will not add any value to this use of animals? We have done the analysis of these the compounds that were submitted by pharmaceutical industry with 5 regulatory agencies which have an exchange of confidentiality. First, the first partners of ICH, EMA, PMDA, and FDA, but later on we also Health Canada and the Swiss Agency for Therapeutics have been included in this test. And you see a picture of the group when we were together with the pharmaceutical industry in Athens when we met for the last time. The prospective evaluation study led to an outcome that it is a tryout with products in the real world, and that's an important one, not only theoretical cases. All sponsors have submitted a carcinogenicity assessment document with a prediction. The regulators decide about the added value. And at an outcome stage of the study, the hypothesis of the prediction has been checked and the added value has been evaluated. And then the guidelines has been changed in August 2022. We have the data sets of 48 carcinogenicity assessment documents, including the 45 study reports, have been published in January 2024 by the paper with Todd Boussier as the first author, and that was an important review of all the data that we have included in this study, and that was published in the Frontiers in Toxicology. And you see the list of authors were the regulatory participants in from the ICH group. And we came to this type of analysis that if the sponsors have their key biologic, pharmacological, and toxicological information, they have to gather the data for the factors to consider. I will show that in a moment. They have to conduct an integrated analysis of the weight of evidence factors. And then we will have these compounds in three different categories. The carcinogenic potential in humans is likely. That's the similar to the category 1 in the hypothesis study. The carcinogenic potential in humans is unlikely. Then why doing such a study? But they have still to do a mouse carc study. And the carcinogenic potential in humans is uncertain. And then we refer to the S1B section 4. We need, in that case, a long-term carcinogenicity study, and if needed, an additional in vivo carcinogenicity study in mice. We have to be aware that in some cases a mouse study may not be appropriate. And those are the factors that have to be evaluated. First, the target biology. So if there is if the target biology is well characterized, it's less likely to do a study. The same is true for the secondary pharmacology. In some cases, there are a few examples where the secondary pharmacology has led to the carcinogenicity. The paper of Frank Sister indicated the relevance of the histopathological data in the chronic studies. If there are no findings of concern or human irrelevant findings, then it's less likely that the study will add value. Of course, genotoxicity is an aspect, but even unequivocal genotoxicity might be important. We should be aware of that. And then the immune modulation. If there is no effect on immune cells or tissues, or even if there is broad immunosuppression in humans, then a study doesn't add value. So both sides of the coin are important. If it is in the middle, and if there is uncertainty, it's shifting to the other side of the figure. What about the reduction of animal use? Based on the analysis we have done, based on the unanimous decisions from the drug regulatory authorities on 3A and B, so studies were which might not have added value, then the reduction might be 12 out of 45, 27%. So there is a number of non-unanimous decisions, and that might be interpreted as the margin to which the percentage of additional approaches can be increased. So companies can do more to support the weight of evidence, can have additional in vitro studies on the properties and the target biology of the compound. And all those questions, we have published that in that paper, all those questions are important in the analysis and the prediction of the value of additional studies. So especially because of an additional interaction with regulatory authorities, requesting further mechanistic data, those aspects can be dealt with when we think about additional studies. So in this table, you see the unanimous 12 and the non-unanimous 12, and that's in fact the non-unanimous 12 is the margin from 27 to 50%. that might be important in the reduction of animal testing. And that's how strong is the estimate of 27%. The data sets of the prospective evaluation study might be biased towards the investigational drugs where sponsors concluded that a 2-year rat study is not warranted. So there might be some bias in what type of compounds have been submitted in this CAD process. The authority is also different in acceptance of uncertainty, the acceptance of risk. That's very true. Without further disclosing any discussion of identity of authorities, everybody feels that there are differences between authorities. Some authorities are more conservative than others. If you want to market it all over the world, you have to consider that you take a risk if you do not study. However, it's also a matter of interaction with the relevant authorities. If you have a good interaction with the in time, whether to or not to conduct such a study on the cost of thousands of animals. So also the ICH is aware of the process. How should there be a follow-up? The EMA has established an operational expert group on carcinogenicity testing to support the non-clinical working party as well as the scientific advice working party. From the Medicines Evaluation Board, Chris Season is my colleague. Chris Season is member of this group. She was also involved in studies in the University of Leiden on carcinogenicity. But also the ICH has established an implementation working group to inform the regulatory parties and to maintain consistency in the application of the weight of evidence approach. I am not sure at this very moment whether the implementation working group has started the work. A few months ago, they were waiting, and I know that the FDA members in the S1 group have left the FDA since the Trump administration. So what is the current status? I have heard the presentation, a webinar to the American College of Toxicology in December 24. The EMA has received 16 requests and all were agreed not to conduct a 2-year rat study. The FDA has received 19 requests, for which 2 are likely to be carcinogenic, 17 requested as unlikely. And then, 8 of them were agreed and 9 disagreed. As far as I know, the EMA cases are presumed not to be the same as for the FDA. There might be some overlap. Nobody knows what is the overlap, except for the group members. So it illustrates the differences in the authorities, but it's not one-to-one, and that it's not sure that all the requests from the FDA have been submitted also to the EMA. In my own life, I've worked on, I've participated in several groups on this issue. There's a collaborative initiative to establish genomic biomarkers for assessing tumorigenic potential, to reduce the reliance on conventional rodent carcinogenicity studies, so completely in line with the topic for today. It is a publication from Chris Corton, published in 2022, and more papers are on the way, and the group is not yet finished with their work. Together with Bob van der Water, I've worked with to have Britt Duindam as a PhD student in this area, as he has worked on the estrogen receptor alpha pathway reporters in human cancer cells, to learn from them in the risk of how to establish estrogen receptors and cell proliferation. This is the first paper of her thesis, and I think that the estrogen receptors of the estrogens are an important human relevant class for carcinogenicity. So this is a relevant test system in this case of S1B. And in the second paper is also the role of genomics. They can enhance the pharmacological prediction for carcinogenicity. And this is a paper comparing the compounds of this test system with the test systems in the OECD for the estrogen hormones and congeners. This is also published already. More data are on the way, and I think that the estrogen receptor stimulation is one of the main risks relevant for the human situation. As a conclusion, a 12-year effort has resulted in an additional approach to establish the carcinogenic risk of human pharmaceuticals without conducting a 2-year rat study. The decrease in animal usage is estimated to be 27%, based on the dataset in the prospective evaluation study. This percentage might be enhanced when the industry will use new methodologies to predict carcinogenic mechanisms, such as genomic approaches. Thank you very much for your attention.
Thank you very much, Jan Willem, for this very interesting and very comprehensive presentation. I think you you describe all the process to arrive at this point with the first to ask for the second species, the mice or not the mice, and then all the biopharmaceutical issues, and then the revision of S1 guideline. So I have a question there. Our first one is from Felipe Vidal. In your opinion, what is lacking for the complete replacement of animal testing in the toxicological assessment of pharmaceutical focusing on carcinogenicity? So the question is one day we will have no animal testing, and what is your opinion? What is lacking?
Yeah, that's a good question. Thank you. We have to be aware that in this process of IGHS1, the starting point is in fact the target biology, as well as a 6 months in vivo study. So is there room to think about no animal use? Thus far, no. We have to be aware that we still need a 6 months tryout in rodents, but yeah, the tryout is in much less animals than 50. The group size in a rodent toxicology study is 10 males, 10 females, or maybe a bit more for 6 months. But you see that the number of animals is much lower. I have at this moment, no other solution than that. We have at this moment, no replacement of the chronic tox study. Thank you.
And Felipe has a complementary question. With beyond validation, what else is needed for a new approach, animal-free, to be considered suitable for assessment of carcinogenicity at the regulator level in the EU. So there is a place for new approach.
Yeah, what is the place for new animal models? I think that
Animal free. He was asking for animal free. Do we?
animal free? Yeah, I
Animal
think
free
that
approach.
the focus is on the target biology. Is the target biology involved in is leading to cell proliferation, and the cell proliferation usually, although not strictly spoken, is cell proliferation not sufficient, but cell proliferation will also fix the spontaneous mutations, or the mutations in another way that is introduced in the cell during time. That aspect is not really expelled out in the system. There are some researchers working on that. I know Barbara Parsons was involved in this hypothesis. The number of mutations is very important. Yes, I think so. But can we test that before? That's not a question.
Okay, thank you. I've got a question from Undergura. So she said it's a basic question about the testing of a non-genotoxic carcinogen, because usually in a carcinogenic process, I mean, there is initiation for chemical initiation, promotion, progression, and she wanted to know how the carcinogenic potential of a non-genotoxic chemical can be assessed in such assay with a rodent bioassay, because sometimes this question is asked, how rodent bioassay are performed to test non-genotoxic carcinogen, since the chemical is not mutagenic.
Difficult to see the point of this question. What I understand in this question is, can we give the safety of a compound, establish the safety of a compound based on the other type of data not being in a rodent 2-year study. As I was involved also in the groups working on the carcinogenic potential for other chemicals. In other chemicals, the regulatory environment is establishing the safety on the basis of no effect levels and so on. And that's not the case or hardly the case in human pharmaceutical area. So it's difficult to indicate in the human pharmaceutical area what is the safe level. In most of the cases, the target biology defines the risk. And if the target biology is relevant in the exposure and has a risk of carcinogenicity, then the target biology is the most important factor, and that's not a quantitative but a qualitative outcome. Is that a question?
I think I can complete also. It means, for example, if you have a compound inducing proliferation of cells, okay, then you have it's target biology, and so the process is that proliferation will provoke the fixation of spontaneous mutation.
Yeah.
So it's target biology, but at the same time you induce mutation due to the spontaneous mutation that are not repaired due to increased proliferation. There's a question from David Guignard. Do you think that the addition of genomic biomarkers in the rat and mouse studies might help to predict the carcinogenic properties of a compound and so to reduce the number of animals per group?
Yeah, that's a very difficult one. Um, do I expect? I hope so that we, on the basis of genomics, we can predict what is the. In fact, what we do with genomics is analyzing the target biology. We can hardly predict the tumors based on that. There is genomic testing is done with even done in vivo, not only in vitro. With for example, the EPA is working with 5 daily, so for 5 days administration, and then looking at the genomics. But it's in fact focusing on the target biology rather than on tumors. Of course, the genomics might lead to the involvement of some important genes, oncogenes, but that has to be established as yet. And yeah, my difficulty with the HASI project is that it is focusing on rat liver because we know so much about rat liver carcinogenicity. But yes, rat liver is hardly relevant for human risk, and yeah, that's one of the weaknesses of the project. The defense of the leading researchers is that the general knowledge will increase, and then the general facts and conclusions will help us to think about what are the relevant factors in genomics, and can we do that also in humans? Yeah, that's we have to wait for the conclusions.
A question. I have a question on the regulatory process. So I think what you present at the end of the presentation is very interesting for the pharma people, because you presented, okay, there have been 17, I think, okay, dossier presented for 16 for EMA and 19 for FDA. You told that it was not the same compound, of course, but in EMA there were 16 agreed, and FDA there were 8 agreed and 9 disagreed, and also there were two compounds that are likely carcinogen. And as I understood that in this decision for the likely carcinogen, which is called category 1, there were two opinions that were non-unanimous, meaning that between the agency, it is right now a difficulty to have some sort of a homogeneous response depending on the compound. I can understand that if a company go to FDA or go to EMA or go to the Japanese authorities, they can have different advice. That's possible. So how to solve this?
Yeah. Good question. No answer.
Thank you.
That's in fact the short answer. That's the reason for this implementation working group. If you read carefully the paper from Boussier, published in January 2024, then you see the discussions on the various cases. There are clear examples where there was no consensus among the 5 authorities. And in many cases, the reasons are that is just giving weight to some effects, whereas others have a general confidence that those other facts are not very important in the whole assessment. So it remains a process of learning, and how to solve that. Yeah, we have advised as a group to have an implementation working group with considering all the cases that have been submitted, and to discuss the differences, as well as the similarities, to learn from each other how things will proceed, and also to publish this type of analysis not only in numbers but also in reasons. So why, and we have to learn from what additional data might be important. In some cases, it's that companies indicate that they have no studies on the metabolites included. In some cases, the background of the proliferative activity is not very clear, or so there are many cases in that all 45 compounds have been discussed in the paper in Frontiers 2024. So that's a learning process.
Yeah, it's very clear. Your answer is very clear, because I mean it will be interesting to see if in some cases, for example, one company go to EMA, one company go to FDA, they have different advice, and then how to solve the issue right away, because it means the company has to make a choice, to conduct the 2 year or not, and the clinical trial is arriving. So find a way that at least the agencies agreed on a common position for the company to progress, because it is a lot of resources and animals involved there. So.
Yeah. So we have done, we started in 2013, and we had not all years 2 or 3 meetings, 2 meetings, but we have physically met each other once a year at least. And then, but of course, because of the COVID, we have more telecoms and video conferences, and that was very effective. So we learned each other, also by meeting monthly in these effects. I'm not sure what will going on in this implementation working group. I hope that they will have regular meetings with sharing the data, sharing the conclusions, to learn from each other, and also to publish that.
Okay, so thank you very much. It's already after 2. It's 2:04. So again, thank you very much, Jan Willem, for this very interesting presentation.
Thank you.
There's a lot of message in the chat saying that thank you for your interesting presentation. And the presentation will be on YouTube if you want then to look at it again. And it's on the YouTube channel of EUROTOX. So have a very nice day, and thank you again, Jan Willem. And see you soon, I hope.
Bye-bye.
Bye.